Hi guys, welcome. Oh wow, that was quick. Hi Alex, how are you? I am good. Hi Hamad, how are you? Look like I'm frozen. I'm just setting up. Literally, I'm just starting. How are things? How are things? Do we have any specific topic areas that we want to cover tonight? Any questions? I'm doing well, thank you. Just waiting for uni to. Ooh, it's such a scary prospect when you just have to wait. Hamid, you're good. I'm really pleased to hear that. Biological membranes, please, from Justin. Can we do hormonal communication? Well, actually, yes, I can. Hi, Tilds. How are you? I saw you'd like some of my videos the other day there. Thank you. Um, right. Yes, I can do the membrane and actually link that with the hormonal communications for Hamad. And yes, if we could do either photosynthesis or respiration as I have a test in 2 weeks, that would be amazing. Alex, you know what? I literally have just, I mean, I don't know if you are interested, but I've just put all my um, resources on my website at pretty much 50% for every booklet. So if you are looking for something for photosynthesis specifically, the one I've just put on, which does transport animals, photosynthesis and plant responses, that one is there. And also I've also got a respiration one available. So I would say looking at those two, yeah, I just put them all at 50% discount for all of them. So if you go over to there, they are on sale right now. Right, let's do hormonal communication, and we're looking at the membrane at the same time. So this is an interesting one. When you do hormonal communication, and specifically, Hamad, what part of hormonal communication are you struggling with? I need to slow myself down. I get a little bit too excited when I'm like, oh, people are speaking to me. This is really cool. Um, and yeah, my muscles run too quickly. Right. So, oh, thank you for the likes, Tilds. I'm calling you Tilds. That's your nickname for me. From me, not for me. Right. Biological membrane. A business supplies steroids and non-steroids have to do with their ability to diffuse depending on cholesterol presence or not. Yeah, it basically is. I mean, the key thing you want to do whenever you're looking at the membrane, I think just the glands and first and second. Right, yeah. I thought that was probably where you wanted to go, but I just want to double check. Right. Oh, Justin, thank you for the likes. Right, here we go. So, Justin, you asked about the phospholipid bilayer, the biological membranes. If you don't know, I am on my other place actually displaying the stuff for you to see. But one of the important things to understand when it comes to biological membranes, and that basically is everything when it's photosynthesis, respiration, excretion, any of that, that is basically all about biological membranes but put into context. Okay, so as long as you're happy with biological membranes and you understand that you have got your phospholipid bilayer, so you can see my phospholipids here, and those phospholipids are creating a barrier. Now, a membrane is a selectively permeable membrane, and the reason it's called a selectively permeable membrane is because it's dependent on which of the proteins are present and how they allow certain substances through. So we know that our phospholipids and our proteins are free to move around within the membrane. And because we know they're free to move around within the membrane, it means they can let some substances through. So it's important that whenever you are trying to get your understanding of this, you think about the fact that if this is my heads, this is my hydrophilic heads, and these are my hydrophobic tails. And so with a fluid mosaic model, the phospholipids are doing this. Well, you can see me if I'm moving a lot, okay? So now imagine my head is the protein embedded in it. Well, I can move my head too, as well as them moving around, because the phospholipids and the proteins are free to move around. So if you understand that we've created a barrier, that we've made a selectively permeable barrier, we therefore need to understand that there are certain molecules that can get through and molecules that can't get through. And it's really dependent on which of those molecules are getting through as to how they're going to affect what happens within the cell. So with hormonal communication then, let's start with the kidneys. Actually, let's start above the kidneys, but just remind ourselves. In excretion, you learn about the kidneys, you learn about the ureter, you learn about the bladder, you learn about the urethra. These are our filtering system that is going to ensure that we don't have any excess salt, that we don't have any excess water, that we get rid of our toxic waste, our urea. So basically, the liver's role of deamination, breaking down excess amino acids, producing ammonia then through the ornithine cycle being converted into urea, has to be excreted, because if it's kept at a high concentration, it will become a toxic substance. So what we have to understand then is, on top of the kidneys are the adrenal glands. So the adrenal gland is a gland which is going to produce through the process. The kidneys do involve ADH. I'll come back to ADH, Tills. They involve the response to ADH, but ADH is actually produced by the hypothalamus, stored in the pituitary gland and secreted from the pituitary gland. So it's still linking it to the hormonal system. But let's first work on the adrenal gland, because actually, Hamad, you've got the stuff on the first messenger and second messenger, and then I'll link it to the ADH, which is also first messenger, second messenger. So the adrenal gland, you have to know about the cortex, which is around the outside, the fact that the cortex is going to release vital hormones. Now, cortisol is one of the first ones you have to know. So cortisol is a hormone that is about regulating metabolism. What is metabolism? Metabolism is about your metabolic processes, all of the metabolic processes that are occurring within an organism. So if we've got the regulating the metabolism, it can up-regulate it, make it happen at a faster rate, or it can slow it down, make it happen at a slower rate. So what are we talking about? Things like, if it's going to affect the metabolism, it could also result in there being a need for regulating the blood pressure. So when you have your stress hormone, as they call it, cortisol, this is going to regulate that blood pressure, increasing it when it needs to be increasing it, or decreasing when it needs to decrease it. So this is a hormone that is going to regulate metabolism, regulate blood pressure, regulate your immune response, and suppress your inflammatory response. So this is an interesting one, because as you get to a certain age, for us that are over the age of 21, clearly not, a bit older than that, but the level of cortisol actually has an effect on whether you store, for example, your glucose, convert it into fats, fatty acids, and then that gets stored as lipids in your adipose tissue. So people who have high cortisol levels actually end up with a storage of lipids in places where they don't want to store it. They end up with bloating because they end up with more fluid being retained, lots of things like that. So cortisol is a vital hormone which is released from the cortex. So I always try to use my alphabet to help me, reminding me. So cortex, cortisol, kind of makes sense. Now this next one doesn't really have a link to it, but you just have to know it's aldosterone. I'll talk about how the aldosterone works. Now, aldosterone is also there for controlling your blood pressure. And it does that by maintaining your blood salt and water concentration. So therefore, it affects your kidneys. So although the adrenal gland is going to produce cortisol, which is going to be secreted out into the bloodstream and circulating all over, aldosterone, interestingly, when it's secreted out, its target organ is actually a close by organ. So you should know that a hormone is something that is released from a gland, and it's released from the gland through the process of exocytosis. It is secreted into the bloodstream. The heart is pumping in a closed circulatory system, so it is being circulated around, and then if it is going to bind to receptors at the target organ, it will then cause a response to occur at that target organ. So a hormone is secreted from one place, the gland. It is then going to bind to a receptor in another place, wherever that may be. Thank you for the likes, Justin. Now, the other place you need to know when it comes to the adrenal gland is the medulla. So the medulla is non-essential hormones, although technically I'd say they feel like they're more essential, but they're classed as non-essential hormones. But they are involved in keeping us alive. So adrenaline is our hormone which is secreted for giving us the animal response of the fight or flight response. So the adrenal gland, the middle section, the medulla, secretes this adrenaline. Now we know adrenaline, if I've got right now I've got adrenaline pumping, it's being secreted, but this comes through excitement. I love and I'm passionate about biology, and sometimes that can result in too much adrenaline being secreted, which is an interesting one, because what that's then doing is that adrenaline is going to increase my heart rate. So if I was to sit here and actually count my heart rate right now, so what I'd be doing is I would be measuring, and I can feel it, it's like literally go do 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 do, kind of like how I'm talking. So my heart has increased. So the adrenaline has actually been secreted from my adrenal gland, and yet my heart rate is increasing. So therefore the adrenaline has been secreted, is transported in the bloodstream, and it is then going to bind to receptors which are in my heart. And it actually binds to receptors in the sinoatrial node. So they have got the receptors that are complementary to adrenaline. What else do I get? Well, rapid increase in my blood sugar levels. So what's going to happen is when I have adrenaline pumping around my body, I am going to either fight or flight. We're not talking about freeze right now. So fight or flight, because that's what you need to know for animal response. I'm going to either fight whatever that is that's causing me to be to the point of needing to be alert, or I'm going to run away. Well, what do I need to be able to do that? I need ATP. ATP comes from the breakdown of glucose. So therefore, I need to rapidly increase my blood glucose levels so that the circulating blood sugars are able to get to, for example, my brain cells, so I am alert and able to move to a safer place, or to my muscle cells, so I've got enough energy so that I can actually do the contractions. And if you've done the animal response and muscles, you'll know that the ATP is actually needed to break those cross bridges. Or it's because I am needing to do active transport of molecules from different places. So adrenaline is going to result in this rapid increase in my blood sugar levels so that it can then be used for respiration. So how does it rapidly... Kashmir tomorrow. Oh, welcome from Kashmir, and good luck in your biology exam tomorrow. How does it actually rapidly increase your blood glucose levels? Well, it increases it by signaling, because remember we're talking about a hormone. A hormone is a messenger. The cells are what do the response to that message that they've been told. So the adrenaline will bind to the cell, and when it binds the cell, that will stimulate the cell to produce the enzymes, for example, or activate those enzymes. We'll talk about that in a minute. That activation of the enzymes for resulting in glycogenolysis. What is glycogenolysis? Well, glycogen is the polysaccharide. So we've got the glycogen, which is a polysaccharide. That polysaccharide, which is made up of 1 to 4 and 1 to 6 glycosidic bonds. So what do we have? We've got the polysaccharide 1 to 4 and 1 to 6 glycosidic bonds. So we have lots of ends that can quickly be hydrolyzed down to release the glucose molecules so that they can then be moved out of the cell into the bloodstream so they can be circulated. The other one we have is noradrenaline. Does it cause glyconeogenesis? So glyconeogenesis is where we're making glucose from the breakdown of amino acids or from the breakdown of fatty acids. So yes, it could stimulate that if that was required. If there wasn't enough glycogen left, then it would look to break down other substances to produce the glucose. So that would be a process which would be if there weren't any available, but yes, it would be. What's noradrenaline going to do? Noradrenaline, released in response to stress, increases your heart rate, widens your pupils. Well, what causes your pupils are just basically the gap in your eye where the light is allowed to pass through. Behind your iris, what you actually have are muscles, 2 types of muscles. And what will happen is those muscles will actually be either signaled to relax or signaled to contract. And because your muscles actually work antagonistically, it's really fascinating that you have muscles that will contract and that will cause the widening of the pupils, but the other muscles have to relax. And then the opposite way around, if you want to constrict your pupils, you've got muscles that need to contract to cause the constriction and other muscles that need to relax that allow for this constriction. So in this case, because we are doing the fight or flight response, noradrenaline is going to widen our pupils. That's going to mean more light can get into the back of our eye. Now, if you're doing AQA, you need to know about the structure of the eye, and you need to know about the retina at the back and the different types of receptors. Hamad, you're talking about the orbital muscles. We're actually not looking at orbital muscles. You're looking at the ciliary muscles, which are going to pull the lens. But what I'm actually talking about is the circular and longitudinal muscles, which are behind the iris. So be careful on the types of muscles that you are talking about, because there's so many different ones. So you've got your ciliary muscles, you've got your circular and radial muscles behind the iris, but you've also got orbital muscles which allow your eyes to move around. So you need to be careful what you mean by the muscles. In the exam, they don't want you to go into that detail. Now, if you're at university or if you're going on to do that for a specialism, then that's completely different. But for A level, you just need to know about the constriction and relaxation of the muscles. Thank you. Wow. Where did I get 1.2k likes? Thank you guys. That's really lovely. Widening of airways. This is important again. This is linking. Everything in biology comes together. In biology, when you're talking about the airways, when you're talking about your trachea going down to your bronchi, going down to your bronchioles and ending your alveoli, you should know, and there's me, oh, here it is. I always have a bubble because I use my hair as my stimming thing. But yeah, if you understand that as your lungs, as your rib cage moves up and out, what's going to happen is that's going to pull the lungs with it. And when it pulls the lungs with it, what you get is this expansion of the alveoli. You get this expansion of the bronchioles, of the bronchi, and what you can have is the elastin fibers can bring it back down to its original position. Well, if we are going to release the hormone noradrenaline, that is going to bind to those muscles, smooth muscles in the bronchi, in the bronchioles, causing them to contract, and that's going to open the airways more so that more air can get in, so we can get more oxygen for aerobic respiration. Because remember, fight or flight, we're going to have to either fight whatever the danger is or run away. So what we've got is that these are coming together, allowing for us maximizing our survival response. And then you have actually the opposite side of things, narrowing blood vessels. So we're going to constrict in some places and we're going to dilate in other places because we want to direct the blood flow to where it needs to go. We need it to go to the brain. We need it to go to the muscles to help us get that strength to fight or to run away because some tigers come in here. Or in the case you're watching a TV screen and you're like, happily, la la, this is lovely. And then you get that rah thing in your face. And you're like, what is that? That would be when noradrenaline has been secreted. And that has resulted in you having all these different things happen. Now, I remember, I'm going to give you a little anecdote of when this happened to me. So I remember when I was 18, I was in a car accident. Now, I was very lucky. The car accident didn't result in too many injuries, but I was actually able to get to work. I was on my way to work. I worked in a nightclub at the time, so it was quite late at night. And I was on my way to work and I got in a car accident and the police had to come and they helped fix the car so I could drive it. I mean, it was a write-off, but not to the extent of that I couldn't still drive it at the time that I was driving to work. Anyway, the reason I'm telling the story, my adrenaline was obviously released, noradrenaline was also released, and I was able to get to work. And when I got to work, straight after I got there, the minute I'd got past the haze of, oh my gosh, this has just happened, my energy levels plummeted. They completely plummeted. And that's because these hormones had caused a response in my body that had broken down lots of the glucose, and the glucose had then been used for respiration. So my sugar levels were so low that if you've ever had some form of situation that's happened, the best thing you can have afterwards, which I still remember to this day having it, is a cup of tea with lots of sugar in it. And it was just a way of calming my nerves down. It was a way of me getting some sugar back into my bloodstream to raise it up quite quickly, because I had used the glucose that had been secreted into my, not secreted is not the right word, but had been moved into my bloodstream. I'd used it, and that was therefore a way in which I could get that level back up before the hormone glucagon was needed, because I'd obviously depleted my resources, so I needed to get sugar into me so that I was at a level that was manageable, which I think is quite interesting. Anyway, here we go. Let's talk about these hormones then. So there's different types of hormones. You've got your, I've got to move this around because of my sign. You've got your non-steroid hormones. Now, these are important to understand. If it's a non-steroid hormone, we're saying it is not able to cross the phospholipid bilayer. Because it's non-steroid, it is going to be prevented from getting across. One, it may be too big. Two, it may be polar. It is a non-steroid hormone. It's too big. It's not able to cross the membrane. So we're talking about insulin, adrenaline, noradrenaline, glucagon, all these ones that I was just talking about. Now, follicle stimulating hormone and luteinizing hormone, you learn in GCSE. You don't have to go into any more detail for these ones. However, it's important to note they could ask a question about it. But if they do, somewhere in that stem of the question, they will have mentioned that it is a non-steroid hormone, or they will have mentioned that it cannot cross the phospholipid bilayer. So if it cannot cross the phospholipid bilayer, it has to be a non-steroid hormone. So therefore it has to be hydrophilic. So we want to try and use those words to maximize our grade. If it can't get across the phospholipid bilayer, it is hydrophilic. It therefore needs to attach to a receptor. So if the adrenaline is going to attach to my receptor, it's going to form a receptor hormone complex. Remember, my receptor could be a protein in the membrane. This is going to bind to it, and they're going to form this complex. So you know about your excuse me, antigen antibody complex. You know about your enzyme substrate complex. Well, this is just a receptor hormone complex. So make sure whenever you've got those two things coming together that you've looked at what it is that's been named. Is it an antibody antigen? Is it an antibody receptor, i.e. an opsinin? Is it a hormone and a receptor? What are the two things that are coming together? They will need to be fusing together, so therefore they must be complementary. What caused the complementary shape of them? Well, let me think. It's a protein. The primary structure is going to be the sequence of amino acids. Then we have the secondary structure. We've got those hydrogen bonds between the carboxyl group and the amine group, giving us our alpha helix, giving us our beta pleated sheet. Then we have our tertiary structure where it's going to fold and bend, and that folding and bending is because of the interactions of these R groups that are in the primary structure. The way in which it's folding and bending, is it ionic bonds? Is it hydrogen bonds? Is it the disulfide bridges? Is it the hydrophobic hydrophilic interactions? Whatever it is that's caused the folding, this tertiary structure, that has made it complementary in shape to the hormone that it's going to bind with. So the receptor has got a very specific shape, which is specific to the hormone in which it is going to respond to. So it's important that you understand that they could ask you about protein structure here, but caveat it with we're looking at the hormonal system when they're really actually asking you about module 2. Do you know that? Do you understand that? So when we have this attachment of the hormone and the receptor together, what's going to happen is that's going to cause the changing of shape, and that changing of shape is going to activate the adenylyl cyclase. Now, for some of you, your teachers will have gone into talk about the G protein and talking about how the G protein becomes activated to activate the adenylyl cyclase. For your exam, you don't need to worry about the G protein. You won't be penalized for talking about the G protein. However, they want you to talk about the adenylyl cyclase. They also want you to be specific in how you spell it. Please, please, please note, if it's written in the specification, the way that it's written is what they're expecting you to write in the exam. You will be surprised how much they are penalizing you for not writing key words that are written in specification in the right way. So do make sure you look at that. So adenylyl cyclase looks weird, but that is exactly how it should be written for the actual exam. Okay, so what's happened? We had our primary messenger, which was our adrenaline. So this was what's known as my primary messenger. That has then bound to the receptor, and that has then activated this enzyme, adenylyl cyclase, and that adenylyl cyclase has then broken ATP down, it's hydrolyzed adenosine triphosphate into cyclic AMP, adenosine monophosphate. So what we have is this cyclic AMP, so the C stands for the cyclic, and then you've got adenosine monophosphate. Now, just to be, oh, crumbs, let's try that again. Let me go out a minute. There we go. Let's zoom in. Just to be clear, when you look at my diagram here, you can see that I've got my adenine nitrogenous base. I've got my ribose sugar because I've got my 2 hydroxyl groups, especially carbon number 2. So it's important that you're looking at these. Carbon number 2 is where we're looking to see if it's a DNA or if it's an RNA. Well, because we're talking about adenosine triphosphate, we know that that is a ribose nucleotide, and then it has tri, meaning 3 phosphates. Well, if we come over here, what we can see is we've got cyclic AMP. We've got adenine attached to a ribose sugar, so this is still the adenosine. The only difference is it's a monophosphate, 1 phosphate. So this cyclic adenosine monophosphate, so cyclic AMP, this is the molecule that is going to activate enzymes, and those enzymes are protein kinases. And so what is going to happen is they are going to result in this glycogen, oh, 1 second, go back, glycogen being broken down into glucose. Your teacher mentioned stomatostatin. I have no idea what that is. This is where I'm like, and this is the thing, different teachers have got different points that they will give you, their little stories that are their way of helping you to know what it is that they are teaching. So let me just have a look. So statins is normally something which is used for someone who has high cholesterol, but is a peptide hormone found in the brain, pancreas. Its main role is to inhibit the release of many other hormones. So yeah, so basically somatostatins even is just a non-steroid. So it's an example of a non-steroid one. So it's just a different people do things in or teach things in different ways due to their expertise. Like I didn't know that, which is like, well, why didn't I know that? And it's because my brain can only contain so much. You're very welcome. And that's the thing to note, that just because your teacher said that doesn't mean they're wrong, because they're not. They're clearly not. It's just they have more knowledge in that, whereas I don't. And so what I would say to you is the content that your teachers are teaching you, some of them are them giving you extra bits of information. Is that necessary for the exam? All those examples that I've given you, not all of them are important for the exam. Insulin, oh crumbs, that's not what I wanted to do. Let's try that again. 1 second. Insulin is definitely important for the exam. So insulin is one that's named in hormonal communication. Adrenaline is one that's named in animal response. Noradrenaline, you will learn more about that as a neurotransmitter as well as the hormone. Glucagon is one that you need to know. Now, the other two I've named, you don't need to know because they're not named in your specification. So just be aware your teachers will always give you more because that's what we want to do. So I would say, do you need to know the somatostatins? No, you don't need to know it as a named example. However, that doesn't mean they won't give you an exam question on it. And if they do, it will be in the stem of the question, information about it that you need to then apply your knowledge to it. So the fact that it is a hormone, which is a peptide hormone, means it's non-steroid hormone. It would need to bind to a receptor. So they would expect you to know that knowledge. Yes, noradrenaline is a neurotransmitter in the sympathetic nervous system. You're absolutely right. If there is time, could we briefly overview DNA replication and need a bit of help for the phosphorylation of nucleotides replication fork? Reminds me what exam board you're doing, because the fact that they've gone into that amount of detail would make me think you're IB or maybe CIE A-level. I'm so stressed. First year of uni biology is nothing like, oh, I'm sorry, James. That's not fun to hear. When water moves out of the collecting duct into the medulla, what process is that called? When water moves out of the collecting duct into the medulla, you are doing selective reabsorption on that one. AQA, but my teacher said it would be really good for the essay to have outside knowledge. Oh, okay. Fair enough. That's fine. I mean, with that one, I'll come back to this. Let me finish the hormones. And then if I do have time, I'll talk you through that. But actually, it's not as difficult as you think it is right now. I'll explain that. I've never heard of it. James, come on. You've never heard of a kidney nephron before? Really? That's a bit worrying. If you did advanced higher in Scotland, did they not do excretion in the kidneys? Check out my video, James. Don't worry about it. Check out my video. Excretion. What is excretion? 5.1.3, I want to say. Find my playlist, which is excretion. I've got videos on the whole thing of the kidneys. Isn't it? Oh, don't worry, James. Honestly, check out my videos on here or on my channel. Actually, on my channel is probably easier, but they're there for you. So don't stress out too much. Okay. Steroid hormones. If we go the opposite side of things, so we were looking at non-steroid hormones. Oh, I just got confused. I was like, what is that? I went and got my vaccination today. That's my little plaster from vaccination. I forgot. I had one on each arm. So I'm going to be building up my B cells and my plasma cells. I was going to do a video on it today, but I haven't had time. And thinking about, antibodies. It's going to take 2 weeks for my antibody level to get high enough. So stay away from me if you've got the flu. Anyway, steroid hormones. What have we got? Well, if we're talking about steroid hormones, remember that aldosterone I talked about? Do you remember that cortisol I talked about? Other ones, again, estrogen, testosterone, progesterone, they're hormones which are steroid hormones. Now, if they're steroid hormones, it means they are non-polar, which means that they are lipid soluble. They can pass straight through the phospholipid bilayer. Now, it's important to remember the hormone needs to bind to a receptor. So either the receptor is on the phospholipid bilayer because the hormone can't get through, or the receptor might be in the cytoplasm, or it might be in the nucleus. So it depends on the hormone. But if the hormone binds to the receptor, we get a hormone receptor complex. Then what's going to happen is that is then going to act as a transcription factor. So for those of you who have done module 6.1.1 cellular control, this should be making sense to you. A transcription factor is something that's either going to promote or inhibit RNA polymerase being able to bind and transcription occurring. So in this case, I'm saying that that hormone's got through, it's bound to the receptor, We've got a hormone receptor complex that's now a transcription factor. It has promoted the RNA polymerase is able to bind, transcription has occurred. I have my mRNA. My pre-mRNA needs to be spliced down into my mature mRNA, and then my mature mRNA is able to leave the nuclear pore, bind to the ribosome. When it binds the ribosome, it can then be read so that the correct primary structure of the amino acids is put together to form whatever the necessary protein is that that hormone is signaling for it to occur. So this is where you have to understand it all interlinks together. Is there a thing called reverse? Yes, there is. Reverse transcriptase is really important. If you have mRNA and you want to get complementary DNA, what you need is a enzyme called reverse transcriptase. So when you look at genetic engineering, you should learn about reverse transcriptase. How you get the gene is one of two ways in which you can get the gene, which is the desired gene. But viruses are really interesting, like the HIV virus. They actually have the protein there. They have reverse transcriptase already inside their protein coat, their capsid, ready so that when they get in, they can quickly convert that from the mRNA to complementary DNA, and then that gets inserted into the DNA strand. It's really, really clever and interesting. It's fascinating, the stuff that they need to do. Right, I forgot. What was I going to do? Oh, I was going to... Hamad, has that helped with your question on the hormonal side of things about the gland and what they do? And I just want to... Give me 1 second. 1 second, 1 second, 1 second. What do I want to bring in? Think, Christine, I want to bring in the replication, DNA replication. Joe, I'm just going to draw. I'm going to draw it really quickly for you because sometimes that helps. So you know that DNA, this is, do not judge my diagrams, okay? So this is me doing it really quickly for you. Da, 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 da. So there's my DNA. We know my DNA is double helixed. La 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 la. Double helixed. Okay, so what you should know is, here we go. It's going to look really dodgy, but we'll go with it. So here you were talking about replication forks. So you know that you have got, I'm going to do this very basic, helicase, DNA helicase, which is going to break the hydrogen bonds. So when DNA helicase breaks the hydrogen bonds, what you end up with is what's known as a replication fork. So the fork, in the fact that it's opening, this has helped a lot. Oh, good. Also, just to confirm, what do I need to know about the pancreas? Oh, that one. The pancreas, you need to know about quite a lot about the pancreas, to be honest. The islets of Langerhans, you need to know about the release, the secretion of the insulin. So you need to know about all the things that happen in there about glucose going in, respiration, the potassium channel shutting, hyperpolarization of the membrane, and then calcium channels, etc. Hamad, if you're not sure on that one, you definitely need to check out my video. I've got 2 videos on it, one on the histology of it and the other one on the secretion of insulin. So hopefully that will help. It really is a lot. Chidima, I would agree with you. It is so much. It's scary. But what you have to do is take a breath to know that it will come together. Right now, this is a scary point in year 12 because you're still learning more and more year 13, sorry. You're still learning more and more content, and you're just like, there's just too much. I don't understand. How can there be more? But actually, if you get a solid foundation of module 2, if you take the time to do it again and again and again, it should slowly start to slot into place. I'm not going to lie to you, it's not going to be easy. It will be challenging, but you need to just keep going, push through it. When you've got a jigsaw puzzle, it just looks like a whole load of random stuff, and then all of a sudden it then turns into, oh, those 5 bits go together. Oh, look, I can kind of see the picture now. Oh, hold on, that goes over there. Those bits go together. Oh, this is making more sense now. I can actually see the picture. That's what happens, but different people have it at different points in year 13. It will come, I promise you. I promise you. Honestly, speak to Tony. Ask her how she felt this time last year when she was in Yearboat. I promise you, she was saying the same stuff. So was Amna. Go and ask them. I promise you, they will tell you the same stuff. It's tough, but you've just got to keep going. You have absolutely got this. Got this? Ugh, I can't even speak. Right. Back to this replication fork. So the replication fork is basically the helicase moving in opposite directions. Now, this is more if you're talking about prokaryotic organisms, because if you think about the prokaryotic organisms, they've got that circular DNA. Do you often get flustered, nervous when teaching a full class, and can they tell? Well, if you look, I go really red and blotchy. So it's quite interesting. I don't get flustered and nervous. I used to be director of student progress, and I used to do assemblies, and I used to teach like 270. I used to do assemblies 270 every day. Like, it doesn't bother me about the big people, like the big crowds. What happens is, interestingly, what worries me the most is, like, I would go red and the kids would be like, oh, she's really red, and I'm like, yeah, and that is my body responding. But I'm older now. When I first started teaching, absolutely, I hated a student coming into my lesson and be like, I'm going to be a doctor. And I was like, oh no, they're going to know more than me. I have imposter syndrome, like, all the time. Like, and I've been teaching this for 20 years. I have a master's from Cambridge. I've got 2 degrees from postgrads from Cambridge, and a biology undergrad degree from Dundee. But still, I get this point where I'm like, I don't know what I'm doing. I don't know what I'm doing. Well, I do, but I don't. I'm just being me, and if you like it and you get something out of it, then I love it. Please like, share, buy my resources, help me do this more. Like, this is the thing. Yes, there are times I will come away from my sessions and be like, oh, that went horrible. I hated that. How bad was that? And yet my bad days are still really good, if that makes sense. And so it's so hard because you feel like you're being judged and you're the person in front of the lesson. I have never forgot, like, there's so many things that I have done in my teaching career that have stuck with me. Like, my first ever training, I said an insect wasn't an animal because I was so stressed, and I was like, ah! And I made a mistake, but it was okay because then I corrected myself. The problem you have as a teacher, if you don't pick up on your misconceptions, or if you don't correct when you make a mistake, your students go off and have those misconceptions, and they go off and they then replicate them in the exam, and that's really rough. What is the microorganism for measles? Sneezing and coughing. Do you know what? I have a friend who, in his university degree, was asked, what do you call the landmass when it was all together, which is Pangea? And he was like, it's Atlantis, because he was like, I have no idea. We all make mistakes. It's really interesting. Everybody makes mistakes. Honestly, you will be surprised how many people will make mistakes. And if people say they don't, they're lying. Honestly, big up Dundee, I love that. I know, I used to work in a nightclub there. The nightclub that I went to after my car accident, Fat Sam's, I haven't been in so many years, but that was my life for, well, from when I was 18 until I left to come down to Cambridge. And I loved it there. I loved it. And then I came to Cambridge, and I loved Cambridge more because it doesn't rain as much here, although it has lately. Taylor, thank you so much for the likes. Nathan, thank you so much for the likes. I do really appreciate it. Right, back to this. Would you say your confidence picked up when teaching? No, no, I wouldn't say my confidence picked up when teaching. I'm a performer in the sense that who I am on here is very like, oh, if you saw me, as soon as I finish this, honestly, I'm going to show you, like, I'm not going to do it because I'm not going to finish, although I've got 5 more minutes, but what I do, the minute I put the camera off, the minute I stop this, I'm like, oh. And I just want to crawl up and I don't want to do that. But when I'm teaching, I get this like excitement and I get this enthusiasm and I love it. And the fact that like for the last 3 years, yeah, well, no, yes and no. When I'm in front of people and I feel nervous and I'm not doing biology, then yes, I did fake it. You fake it till you make it. You literally convince yourself you're confident. But for me, I'm neurodivergent, so I basically like my space and I get overstimulated. So the fact I'm in this room and I'm like talking away and this is my safe space, this is my comfort zone. And I used to say like my work, how I was at work and how I was at home were 2 different places because I need that shutdown time. I need that time to just recoup. But it's one of those things that you will get it back. Students can and do pick up when you know and you don't know things. And if you've lost the trust in your students, I'm not going to lie to you, but if you've lost the trust in your students, then that becomes a problem. However, you can get it back. It's okay to have bad days. It's okay. You're human. You're natural. You're a teacher. You're not this entity that knows it all. You can have bad days. I would have days where I'd be like, ah! But it's about what you do with repairing that conflict. So conflict resolution is a massive thing where teachers forget that they need to repair the relationships with their students as well. Students are not just going to sit in your class and trust you and listen to you and take your guidance if at any point you have ridiculed them or if you have belittled them or if you have given them some reason to distrust you. However, if they trust you and you make a mistake, they're actually quite forgiving as well. So you have to realize that it works on both sides. If you're just starting out, then you're just starting out. That's okay, but you need to make sure you're improving and you're learning from the mistakes that you make. Too many times, if we don't improve and we don't make the movements to improving, that's where it becomes a real struggle. And I would say the fact you're reflecting on it right now, the fact that it's affecting you, that says more about you than somebody who just goes, oh well, doesn't matter. Right, I'm not gonna forget how to do this last bit for you. Not even forget, I'm not gonna stop. Phosphorylation of nucleotides. So this one's important. When we teach DNA replication, we talk about these nucleotides and the nucleotides coming along and forming complementary base pairs. All of that is correct. I'm not telling you anything that isn't correct. But what we don't talk about is the fact that the free nucleotides that come and form the hydrogen bonds, they actually have 3 phosphates. They're actually a triphosphate. So they're a deoxyribonucleic oh my word, I can't even do it. They're a DNTP, nucleotide triphosphate. So they are a deoxyribonucleotide triphosphate. And what will happen is they will break off those 2 phosphates. And when they break off those 2 phosphates, that provides the energy for the DNA polymerase as well, binding them together, giving you that phosphodiester bond. So it's an interesting one that it's okay to have the extra information, but be careful that you don't get hung up on that, because what's important in the exam when it comes to that essay, and that 25 marker isn't a small task. It's huge, but the key thing is if you are linking together 3 or more topic areas And you haven't just given generic answers. You've gone into a bit more detail. You've given some really good scientific vocabulary. That is what's going to help you to maximize that grade. Sam, what I'd say to you for DNA transcription, I have a video on it on my channel. You want 2.1.3 nucleic acids and nucleotides playlist. There's one specifically on transcription. The video is there. I'm going to be doing some more. Or the other alternative is on my website. You can buy my nucleic acid nucleotide study guide. I've just put it up for 50% off. So it's on sale right now. So if you go to my website, ukbiotutor.com, you will find all my revision guides are all half price. I feel really disheartened because nobody's seeing the hard work I've put into these, and they are so good. Like, I'm not just saying it, they are so good. So I would say definitely go and have a look there and see if that helps. Chidima, thank you for joining me. I'm sorry that it's been too much. Quick run through of it, please. I can. It's like a minute past my bedtime. I need to go to bed. I have been up since 5 this morning. I'm exhausted. I will be back on here on Wednesday. I'll go live again on Wednesday. So please do come and join me then at 9 o'clock on Wednesday. And have a good evening. I broke your heart. Oh, no. Come on. I didn't break your heart. You can't say that. I've literally been on here for 56 minutes for free. And just because, and I've told you where to go to find the resources. It's not even as if that I haven't answered any of your questions tonight. What's the difference between eukaryotic and prokaryotic? Go check out my 2.1.1 playlist. You'll find it there. Oh, you've only joined 10 minutes ago. Well, there you go. That's the problem. It's okay. I'll be back on here on Monday at 9 o'clock. But if you check out my channel, the whole year 12 and 13 course, it's all there. All right, babes. Don't have time for playlists. Oh, shocking. Oh, never mind. I guess I can't help you then. Is your content specifically for OCR A? Yes, it is. It is OCR A. It is from the 2023. I spent 6 months last year building the YouTube channel. And so it's all there. The video, how long ago did I post my transcription one? Transcription would have been quite early on, but it doesn't mean it's not. What do you mean you really can't find? Oh, on here. Yeah, you're not going to find it on here. Go to my YouTube channel. It's in a playlist specifically, which says 2.1.3 nucleic acids and nucleotides. What's the difference between AQA and OCR? AQA and OCR are just different ways in which they assess the content. There's not as much to learn for AQA. However, they do have the 25 marker. They've also got the big section where you have to like take they've written like 15 lines worth, and on line 5 it says this, and on line 10 it says that. So that's the main reason. I'd say the questions, the short questions on AQA are much easier. OCR's biggest question is a 6 marker, and on that one I would say OCR's got more content, the shorter questions, but they're quite a lot of application. NXLB, wow, your questions are rough, some of them, but I love them because it's like proper biology application, but it's rough. But you don't have to learn as much content. So it just really depends on each of the different exam boards. Using the specification as your cheat sheet, because it will tell you exactly what you need to know. What you also have to do is use your exam board's past papers and mark schemes so you can get into a habit of what will they accept or what won't they accept. That's what I would say. My resources are built for OCR A because there was nothing for me to direct my students to. However, there is like a 75% overlap in all the exam boards, so the content is still relevant. But for example, in AQA you have to know the word photoionization. You don't have to know that in AQA, in OCR, sorry. And things like when you do the linked reaction, you have to call it acetate, whereas we call it acetyl group. So it's just like slight tweaks that they want. You have to know about centrifugation and homogenization, whereas we don't need to know that in OCR. So there's just differences about it that you need to be specific in what you're doing. Right, I am going to go. It's 5 past. I stayed an extra 4 minutes. I could have probably done. No, I couldn't really. Thing is, if I get started, I don't stop. So, guys, thank you for joining me. Have a good evening. And I look forward to seeing you all again with me on Wednesday.